Review of Chapter 9, Cellular Respiration. Cellular respiration is the process of converting energy from food into ATP, adenosine triphosphate. There's a lot of information in this chapter. Make sure you study the notes and more specifically what the notes emphasize. Now, I have the summary equation of this on the board. You also have the summary equation on the bottom of page 164 in your textbook. Now, yes, we can convert carbohydrates, fats, and proteins for energy. They provide the calories in our food. But it is easiest to describe cellular respiration from the glucose point of view, the oxidation of glucose. So on the summary equation, we have a molecule of glucose, C6H12O6. To oxidize this completely, you're going to need 6 molecules of oxygen. That's what it's for. That's why we need oxygen. When it's all said and done, we breathe out the carbon dioxide. Some water molecules are gained, and we have energy. Now, this energy is in the form of two things. Enough energy to phosphorylate 38 ATPs. And of course, heat is left over. Remember the second law of thermodynamics. Energy conversions are never 100% efficient. In this case, we're talking about 40% efficiency. Not too shabby. Now again, ATP is used for all cellular work. Now, what happens is that there are 3 stages of cellular respiration. In other words, this process occurs in 3 stages. If you want to look at page 166, it gives you an overview of cellular respiration. Now, the 3 stages are glycolysis, the citric acid cycle, and oxidative phosphorylation. You'll notice on page 166 that 2 of the 3 occur in the mitochondria. Again, that's what it's for. Now, I'm going to go over these very, very briefly, and I want you to study the notes. Now, glycolysis. The word glycolysis means to split sugar. Okay? So, this happens in the cytoplasmic fluid. It does not happen in the mitochondria. Glycolysis is not aerobic. It is anaerobic. This stage does not require oxygen. So oxygen is not the oxidizer. There is another oxidizer. Okay? And that oxidizer is known as NADH. It's a coenzyme. Now, this partially oxidizes glucose. The word glycolysis means to split sugar. So one glucose is split into 2 pyruvate molecules, and we have a net gain of 2 ATP molecules from the deal. Okay? We actually use 2 ATPs to make 4, but the net gain is 2. So that's glycolysis. Now, the details on glycolysis are on pages 168 and 169. I am not going to make you memorize all of these steps on pages 168 and 169 for exams, okay? But that's what's going on with glycolysis. The second step is the citric acid cycle, pages 170, 171. The citric acid cycle is also known as the Krebs, K-R-E-B-S cycle, or the Krebs citric acid cycle. Now, again, I'm not going to make you memorize all of the molecular steps of the Krebs cycle, but what I do want you to know is that it happens in the mitochondria. And what happens, if you look on page 170, we start off with pyruvate, the end product of glycolysis. We have to convert it to another molecule called acetyl-CoA, which enters the mitochondria. It is acetyl-CoA that enters the citric acid cycle. You lose a molecule of carbon dioxide when you do that. So, acetyl-CoA enters. Most of the energy is still left in this acetyl-CoA. That energy is transferred to these coenzymes, NADH and FADH2. And we have a net gain of 2 more ATPs from our original starting point of glucose. So this acetyl-CoA is completely broken down, and the rest of your carbon dioxide is produced here in the citric acid cycle. The third stage in cellular respiration is the oxidative phosphorylation, also known as the electron transport chain. This occurs in the inner membranes of the mitochondria. This is where most of our ATP is produced. Some 36 molecules of ATP are produced from this third stage. This is the stage that actually uses oxygen. So what we have is a chain reaction involving a series of reactions transferring electrons from molecule to molecule to molecule all the way to oxygen. Now what this is doing is that this is generating a steep hydrogen ion gradient, a steep proton gradient across the inner mitochondrial membrane. Now these protons do not easily diffuse through the phospholipid bilayer of the membrane. So there is a molecule known as ATP synthase, in fact I'm on page 174, 175, where as we build up the steep proton gradient, those hydrogen ions, or protons, are going to want to diffuse through that protein. And when it does, it catalyzes the phosphorylation or regeneration of ATP. It works kind of like a hydroelectric dam, where a dam of a river builds a steep gradient of water. And then as the water flows through the dam, it generates electricity. Here we're building a steep hydrogen ion gradient. As it flows through ATP synthase, it generates ATP. Now, a summary of all of this is on page 176. Again, glycolysis does not occur in the mitochondria. The other two do. Citric acid cycle and the electron transport chain. So, glycolysis gives us 2 ATPs. Citric acid cycle gives us 2 more. Oxidative phosphorylation, electron transport, and chemiosmosis. That word chemiosmosis is what I've just said, where you've got the steep proton gradient and it's going through that protein. That's chemiosmosis. Generating 32 to 34 ATPs, giving us a grand total of 38 ATPs from one glucose molecule. Now that's aerobic cellular respiration. There are cells that can live anaerobically. Now, in an anaerobic condition, you can't go through the stages in the mitochondria. You can only use glycolysis. Okay? So what happens with glycolysis, remember that the oxidizer is not oxygen. But you can't run out of that oxidizer either, or that cell can't even produce any ATP at all. So what fermentation is it's the anaerobic alternative. It allows for pyruvate to be broken down further in order to recharge the oxidizer, the NAD, so that glycolysis can occur. Now, fermentation is only about 2% efficient at converting energy into ATP, but in the absence of oxygen, that's the best you're going to do. Now, several forms of fermentation are mentioned. Alcoholic fermentation by brewer's yeast, where pyruvate is ultimately broken down into ethyl alcohol and carbon dioxide. There's also lactic acid fermentation, which can occur in muscles, fatigue muscles, in anaerobic conditions, where lactic acid is produced by converting pyruvate into lactic acid. Now, there's no CO2 in that conversion, fortunately. There's many kinds of fermentation from anaerobic bacteria. Now, a strict anaerobe is an organism that must live in the absence of oxygen. Certain bacteria are strict anaerobes, such as the one that's responsible for botulism. A facultative anaerobe is one that can live with or without oxygen, in which case it can use aerobic or fermentation, brewer's yeast, and many bacteria as well. And then you have strict aerobes. These are organisms that must have oxygen to live, like you and I, and most other organisms as well. Well, that pretty much summarizes this chapter, and there you go.